Hello, everyone. So my name is Shruthi, and I work as a doctoral program manager and postdoctoral fellow at the Center for Wildlife Studies. Welcome to today's webinar, Buzzing Insights, where we'll take a deep dive into a topic that's very close to my heart, Asian honeybees. Compared to its Western counterpart, the Apis mellifera, Asian honeybees haven't received much attention. But in today's webinar, we'll get to know a lot more about them, especially about their foraging behavior, pollination ecology, as well as the hornets that predate upon them. And to talk and discuss about this, we have two very wonderful speakers with us, Professor Vasuki Belavadi and Professor James Nieh, both of whom I know don't need much of an introduction, but I'll still give a brief intro. Professor Belavadi completed his bachelor's in agriculture from the University of Agricultural Sciences in Bangalore and master's in agricultural entomology from the University of Agricultural Sciences in Harvard in 1976 and 1979 respectively. He then returned to the University of Agricultural Sciences, Bangalore to complete his PhD in 1987. He has held multiple positions at several institutes, including the Central Agricultural Research Institute in Port Blair and the University of Agricultural Sciences in Bangalore. It was here in the latter that he superannuated as professor and head of the Department of Entomology in 2018 and continues to serve as the ICAR Emeritus Scientist from 2018 to 2021. He has been a fellow of the Royal Entomological Society, London since 1994, and he has served as a consultant for the Global Pollination Project of FAO from 2010 to 2012, and as a member of the midterm evaluation team for the Global Pollination Project, UNEP, in 2012. It was during one of his stint at the Zonal Agricultural Research Station in Mudigere that he started his work on pollination biology. His initial work was on cardamom pollination that helped in recommending to the farmers regarding the optimal number of bee colonies required for successful pollination in the crops. Later, he continued his interest in other crops as well with the help of his students. Thank you so much for being here, Professor Belavadi. It's a pleasure to have you with us.
Thank you.
Next, Professor James Nieh. Professor Nieh was born in Taiwan and grew up in Southern California. He received his BA at Harvard in 1991 and his PhD from Cornell University in 1997. He subsequently received an NSF NATO postdoctoral fellowship to study at the University of Würzburg in Germany. After this, he received the prestigious Harvard Junior Fellowship. And in 2000, he joined the faculty in the School of Biological Sciences at the University of California, San Diego, where he is a professor in the Department of Ecology, Behavior, and Evolution. He held the Heiligenberg Chair of Neuroethology, was chair of his department, and was elected fellow of the Royal Entomological Society in 2017. He is an associate dean in the School of Biological Sciences. Professor Nieh's interest focuses on bee communication, cognition, and health. He studies many types of social bees, including honeybees, bumblebees, and stingless bees. His lab studies natural and man-made stressors of social bees, and a major part of his work focuses on foraging and communication in honeybees and honeybee health. Thank you so much, Professor Nieh, for accepting our invitation to join the webinar. And I know for you it's a really early time, so thank you very much for joining us. And I really look forward to your talk. So the structure for today's webinar, we'll start with a short presentation by Professor Belavadi, about 15 to 20 minutes, followed by another short presentation by Professor Nieh for 15 to 20 minutes. And then we'll have a general discussion. And the last 5 to 10 minutes will be open for audience Q&A. With that, we'll move on to Professor Belavadi. Over to you, sir.
Okay, thank you. I'll just share my presentation.
Sure. Yeah.
Okay, thank you, Dr. Shruthi, for the nice introduction. Sorry, what did I do?
No worries.
Okay.
I can see your screen. Yeah.
No, I am unable to see the screen. Okay, yeah.
Oh.
Right. I'll be speaking today on the foraging behavior of honeybees. And mainly I'll be concentrating on two crops. One is on cardamom, on which I have worked in the beginning, and also on muskmelon. These are the two crops on which we have done some interesting studies on the foraging behavior of honeybees. Now, before going into the foraging behavior, I'd like to spend a few minutes on the diversity of bees. Diversity of bees in the world, diversity of bees in India, and the diversity of the genus Apis. Now, there are 20,925 species of bees in the world, which have been put under 7 families of bees And in India, we have 755 species under 6 families. We do not have one of the families, that is Ctenotridae, is so far not recorded from India. Now, when it comes to the genus Apis, there are about a dozen species of Apis, and majority of them are found in Asia. For example, Apis andraniformis is found in Bhutan, China, Vietnam, Malaysia, Indonesia, and Philippines. Then Apis cerana and Apis dorsata, of course, they are found in India and Southeast Asia and part in the whole of Asia, cerana. And then Apis florea is found again in India and also Southeast Asia and also a few parts of Northern Africa. Then Apis koshnevicovi is found only in Thailand, Indonesia, and Malaysia. Then Apis laboriosa in Nepal, Bhutan, India, Burma, Laos, and Vietnam. And Apis nigrocincta, Malaysia, Indonesia, and Philippines. And Apis nuluensis is recorded only from Malaysia. Now, compared to this, Apis mellifera is recorded from all over India. It is because of the, introduction of Apis mellifera to various countries by man. So we know that now that almost all species are found in Asia. Are you able to hear me?
Yes, yeah.
Okay. Now, coming to the foraging behavior, the foraging behavior refers to the searching for food. And these actually forage for pollen and nectar. They communicate about the location, and especially using certain dances, round dance and waggle dance, to inform the fellow workers about the distance and direction. And this saves a lot of time and energy because they actually can recruit workers for food collection. So I'm not going to go details about the dance communication and other things because it is a very big subject. Now, I'm going to dwell on the foraging behavior of honeybees on 2 crops. As I said, one is cardamom, Elateria cardamomum, and muskmelon, Cucumis mellow. Cardamom is the queen of spices. It belongs to the family Zingiberaceae. It is native to the Western Ghats of India, cultivated under the shade of trees, and in fact, it is the 3rd costliest spice in the world after saffron and vanilla. It's cultivated in India over 70,000 hectares in 3 southern states, mainly Karnataka, Kerala, and Tamil Nadu. And the production is around 24,500 tons annually. And a lot of cardamom is exported. And in fact, in 2023, 6,500 tons of cardamom was exported, earning $103 million to the country. Now, when you look at the cardamom plant, it gives an impression that it is a cluster of pseudostems. In fact, a single plant may consist of several pseudostems, and that is the reason why we also often call a cardamom plant as a clump of pseudostems or a clump. At the base of each pseudostem, it produces 1 or 2 panicles, and the panicles lie prostrate on the ground, and the flowers are borne on these panicles. Now, in a field of cardamom, in a plantation of cardamom, if a bee enters, if a forager enters, it has to come to the ground level and then visit the flowers around the plant, visiting flowers. So, and most often you see 2 species of bees, Apis cerana and Apis dorsata. These are the major pollinators, but there are also a few non-Apis bees as well. I'll come to that a little later. Now, flowering phenology and floral biology of any crop is important when we are working on the foraging dynamics of the pollinator. The flowering of cardamom starts late in the last week of April, and it continues till November. And anthesis or flower opening is in the early morning hours, 4:30 and 6:30 a.m., between 4:30 and 6:30 a.m. And pollen dehiscence is around 7:30 a.m. And the flower longevity is just for 12 to 15 hours. That means a flower that opens in the morning, it has to get pollinated on the same day before evening. Otherwise, it just falls off. So every day, if you go to the cardamom field, you find new flowers. Every day, it is a new flower that you see. And the flower density is highest in July, August. In the months of July and August, the maximum flowering is seen, and the density per clump of cardamom also is very high. Because if the density is not if the density is very low, that is, if the number of flowers per clump is less than 5, honeybees do not visit. Neither Sarana nor Dorsetta visit these flowers because it is too costly for them to forage on such clumps when the flowers are distributed in a longer area. They have to spend a lot of time flying between plants and all that. So under such circumstances, when the flower density is very low, it is the non-apis bees like Amethylna which are also excellent pollinators of cardamom. Now, the foraging activity depends on nectar availability, and the activity also should coincide with pollen viability and stigma receptivity in the flower. In cardamom, the pollen is viable throughout the day. However, the pollen viability is maximum around 12 noon, between 12 and 1 p.m., 12 p.m. and 1 p.m. And the stigma receptivity is also there throughout the day, but the maximum receptivity is in the afternoon, that is again between 11 and 3 p.m. And continuous nectar production is there in the flowers, and the nectar secretion, the rate of nectar secretion is highest around 11 a.m. and then it comes down, and the bee activity also is high between 11 and 3 p.m. So that is this coincides, the bee activity coincides with the pollen viability and stigma receptivity, and so bees become excellent pollinators of cardamom. Now, as far as the foraging behavior is concerned, each forager makes about 3 to 4 trips every day in a day, and each forager visits 120 to 135 flowers per trip. In each trip, a forager visits 120 to 135 flowers. This is because the Apis cerana has a crop capacity, the stomach capacity of 40 microliters, and to fill its crop, it has to visit so many flowers because in each flower, the bee is getting only about 0.3 microliters of nectar. So it has to visit about 130 flowers to fill its crop. Unless its crop is filled, it will not go back to the nest. And a single flower is also visited several times, and this is also necessary because in a flower of cardamom, in the ovary, there are 27 ovules, and each ovule requires a pollen grain. And with 1 or 2 visits, so many pollen grains may not be deposited. So as the number of visits increase, the number of pollen grains deposited also increase. There is a positive relation with that. And there is also a positive relation with the fruit set. With more number of visits to a single flower, there is higher probability of setting. Then another important feature of the flower of cardamom is that the corolla tube is about 23 millimeters long. The corolla tube is 23 millimeters long, and you will find in the peak flowering season, you will find that nearly 50% of the nectar remains unharvested. It remains in the cardamom. This is because both Apis cerana and Apis dorsata have relatively shorter tongue lengths. The tongue length of Apis cerana is 4.5 millimeters, and Apis dorsata has a 5.5 millimeters tongue. And they cannot exhaust all the nectar that is available in the corolla tube. For example, if this is the corolla tube with full of nectar, a bee sits there and then it can draw nectar only up to about 50% or about 11.5 to 12 millimeters. So when a bee approaches a flower, it hovers over the flower for some time before deciding to land on the flower. While hovering over the flower, it will come to know that that flower has been recently visited by another bee. So there is no nectar available for it. So it leaves and then moves to the next flower. This is a very important thing to know about the landing and departure rules followed by these foraging bees. For example, if the nectar level is below 11 millimeters, usually the bees do not land. And the landing is observed only when the nectar level is beyond 12 or 14 millimeters. So when the nectar level is low, they just hover and then leave that flower and go to the next flower. And if the nectar level is higher, there is more likely chances of landing on the flower. And when a bee approaches a clump, it first lands on the peripheral flower. It first approaches the peripheral flower and then moves to the middle and then to the base of the clump. And similarly, while leaving the clump also, it leaves from the peripheral flower. And a single bee, when it is visiting, it visits invariably in a clockwise or anti-clockwise direction. It moves in clockwise or anti-clockwise direction around the clump. And it will visit the nearest neighboring flower often. And interestingly, if it does not find nectar in the first 3 flowers, it goes to the first flower and it does not find nectar. It goes to the second flower, there is no nectar, and then it goes to the third flower, there is no nectar. Then it leaves that clump. Okay, it goes to the next plant. So this we call it as sampling by the bee. They sample up to 3 flowers to decide whether to stay on the that particular clump, that particular plant, or not, or move to the next plant. So if they are not getting nectar, finding nectar in first 3 flowers, that means they understand that this plant has already been visited by bees very recently. Then the movement rules followed by foragers, we can put it like this: land on the peripheral flower and move in clockwise or anti-clockwise direction, visiting the nearest neighboring flower. Do not land on the flower if nectar is not reachable. If nectar is not available in the first 3 successive flowers, go to the next plant and depart from the peripheral flower to the next plant. So this is about the foraging behavior of bees on cardamom. Next, I'll move on to muskmelon. Muskmelon is andromonaceous. That means there are 2 types of flowers on each plant: hermaphrodite flowers and also staminate flowers. This is the photograph of the staminate and the hermaphrodite flower, which is there on a single plant. And the ratio between the staminate and hermaphrodite flowers is about 18:1. That means for each hermaphrodite flower, there will be 18 staminate flowers. And muskmelon flowers are visited by Apis florea and Apis cerana. Now, pollen is available in both staminate and hermaphrodite flowers. So the question is there any difference in the quantity or quality? Or the question can be, when the hermaphrodite flowers are there, when the pollen is available in the hermaphrodite flower, why the plant should have staminate flowers? So this is a kind of a question that can come to anybody. And when we looked at the quantity of pollen available, we find that staminate flowers have significantly higher number of pollen grains per flower compared to hermaphrodite flowers. It is almost double the number of pollen grains in the than the hermaphrodite flowers. And the next thing is about the quality, the pollen viability. The germination test, when we did, we find that the pollen grains from the staminate flowers germinate better compared to hermaphrodite flowers. Hermaphrodite flower, the pollen from hermaphrodite flower also germinates. But the next thing is the germinated pollen grain the pollen tube does not grow further beyond 2 micrometers in the pollen from hermaphrodite flowers. So this gives an indication that the pollen grain from the hermaphrodite flower may not be fertilizing the ovule. Apparently, pollen from hermaphrodite flowers cannot fertilize any ovule. So we did a field experiment to test this, in which we had 5 treatments. One is for open pollination, then selfing, then bagging, selfing of the hermaphrodite flower, bagging the hermaphrodite flower, then hand pollination with pollen from another hermaphrodite flower, then hand pollination with pollen from staminate flower. And interestingly, we found that percent fruit set was 0 with selfing, bagging, and those with pollen from hermaphrodite flowers, whereas fruit set was good in open pollination and also with the pollen from staminate flowers, which clearly indicates that it is only the pollen from the staminate flowers that are useful for the setting of seeds or setting of the fruit. So what happens if we emasculate the hermaphrodite flower, since it is not needed for the pollination at all? So what we did was we did the emasculation, and interestingly, we found that in emasculated hermaphrodite flowers, the setting was less, and the seed number per fruit was distinctly or significantly less in emasculated hermaphrodite flower. So why this difference in fruit set and seed number was the next question. If pollen from hermaphrodite flowers are not useful for the plant, for the reproduction, what are they for? That was the next question. So next, we wanted to look at, do foragers visit staminate flowers before visiting a hermaphrodite flower? So we found that Apis cerana bees visit invariably about 10 to 12 staminate flowers before visiting a hermaphrodite flower, and Apis florea visits about 8 staminate flowers before visiting a hermaphrodite flower. Okay, so they are visiting large number of many staminate flowers and picking pollen, collecting pollen, and then there is nectar in both the flowers, and they are also collecting pollen from the staminate flowers and also from the hermaphrodite flowers. And the time spent per flower is interesting. Both Apis cerana and Apis florea in emasculated hermaphrodite flowers, they spent least time, very little time compared to normal hermaphrodite flowers. And the number of pollen grains deposited on the stigma of the flowers was also significantly higher in normal hermaphrodite flowers compared to emasculated hermaphrodite flowers. So the thing is, foragers spend more time in hermaphrodite flowers to collect pollen, and the male part of the hermaphrodite flower helps in retaining foragers for a longer time to improve pollination efficiency. I think that's it. And this is my team. Thank you. I'm ready for any questions.
So that was a really beautiful talk. So before we move on to Professor Nieh's talk, I just wanted to ask a real quick question. So I was just wondering if the recruitment within the colony. So when you talked about the cardamom flowers and how sort of very fickle the whole flower nectar is. So how do you see a stark difference in the recruitment process that happens within the colony for these flowers compared to any other food source?
It will be like any other food source only. The recruitment is the same, but individual foragers should take a decision for landing and moving about. So that is the effect. And interestingly, they will be, if a bee is making, say, 5 or 6 trips in a day, it keeps on coming to the same patch. It will not go to a different patch each time. It comes to the same patch because the nectar secretion is continuous. Continuously, it is the nectar is secreted. So they keep coming. They get the nectar. Though when no bee is allowed to visit, when you close the flower, when the corolla tube is completely full, it will have about 4 microliters of nectar, but no bee will ever get that. Okay, so because once a bee visits and then it takes nectar, it goes up to about 11.5 millimeters, and the next bee will get only about 0.3 microliters. So that's how it is. It takes about 15 minutes for this secretion. So that is the time between the visits. Yeah.
Right, I see. Okay. I think Professor Nieh had a question.
Thank you, Professor Belavadi. That was very interesting, particularly about cardamom, which I also love as a spice. But I had a question. Because the corolla tube is longer than the proboscis of the bee, was there originally a different native pollinator that co-evolved with this plant?
No, the thing is, in fact, I have a paper on that. In fact, I can send you that, how they managed to take nectar from up to about 12 millimeters, though their tongue length is only about 4.5 millimeters. Okay, it is some kind of a capillary action by which they try to take that nectar. That's how they take this thing. Yeah.
Thank you.
Yeah.
Interesting. I think we'll have a more general discussion at the end. So we'll move on to. Thank you so much, sir. We'll come back with more questions, but for now, I think we'll go with Professor Nieh's talk. Yeah.
Thank you very much. Okay, so let me go ahead and share my screen. My presentation just vanished. Here we go. Okay, so, well, first of all, I wanted to thank everyone, particularly Sruthi, for your invitation. It's very kind, and what a wonderful opportunity to meet, which is actually a fairly sizable Zoom group I see so far, 45 participants. Okay.
You both are very popular.
Oh, thank you. All right, so I am going to choose a highlighting color. Okay, great. So my talk today is about fear fighting and coevolution between Apis cerana and its horned predators. And I thought this topic would be of interest, particularly because you have a subspecies of Apis cerana indica, that is widely found throughout India. The big picture of my study is that competition, both within and between species, as we see in predator-prey relations, generates dynamic fitness landscapes that influence the evolution in remarkable ways. Now, we don't usually use the term fitness landscape. I'm using it specifically here because although we often now talk about it in terms of a genetic landscape, the concept is actually applicable to this more traditional view of arms races. And the other thing that I wanted to emphasize is that I, like many other bee scientists, find that honeybees are really a magic well, this term from Von Frisch, for understanding evolution. And so the two stories I want to discuss today are referential stop signals and warning to predators, which you actually see here. So you might ask the question, why is this Asian hornet running away and not attacking the bees? The work that I'm describing today was done in collaboration with Professor Kim Tan's lab in Yunnan, China, in Kunming, China. So the first story, predators, competitors, and shouting stop. There will be AI images present in my talk, and they should be fairly obvious, for example, such as this one. So referential communication, we are familiar with in terms of the honeybee waggle dance. It encodes information about the distance and direction to the food source. But such referential communication is found throughout the animal world. So even chickens, although we don't think of them as very smart animals, have alarm calls that change depending on how far away the predator is. Similarly, Mexican chickadees, for example, give higher pitched alarm calls when the danger is more severe or closer by. We have red-fronted lemurs that increase alarm vocalizations, and of course, the charming social mongoose, whose alarm calls also vary acoustically. Of course, in humans, we have a wide variety of ways of communicating danger, and these change depending upon the urgency of the threat. So the question is that previously, our lab identified something called the stop signal. This was known previously as the begging signal. It's thought to be a signal that bees give to get food. But what multiple researchers have shown now is that bees don't get food when they give the signal. What does happen, though, is that the waggle dance tends to stop. And so previously, what I and others have found is that the stop signal stops waggle dancing when conditions of the food source are no longer good, such as when there are too many bees visiting a feeder. If you've ever trained bees to a feeder, you'll have encountered this. Or when bees encounter a predator or attacked at the food source by bees from another colony. I've shown this rather dramatic image to compare. What you see here is Vespa velutina, the smaller Asian hornet, compared to Apis cerana, which it is now attacking on a flower. And we also have Vespa mandarinia. This is one of the famous giant hornets. And here what you see is it's much larger in size. So the question is it perceived by bees as a larger threat? So in our experiments, we follow classic methods of Carl von Frisch. We train and mark Apis cerana at a feeder. And then we present a hornet, which is tethered to a wire. The reason for this is that if the hornet is flying around, it would often flee, and this allows us to experimentally manipulate where the hornet is relative to the colony. Now, Apis cerana is famous for its heat balling behavior. You probably have encountered this before. This is a thermal image from Ono, and what you know is that the bees will actually heat up so that they can either incapacitate or, in extreme cases, kill the predator, the hornet, inside by generating heat. And this takes us back to arms races because, after all, in response, over evolutionary time, the hornet, we believe, has developed very thick armor, and so the honeybee, cleverly enough, comes up with a social strategy of generating a large number of bees to kill it. We'll talk about that more, but I wanted to show you that these bees are capable of defense. And this is now Professor Dong Xiao, who is using an observation colony and a video camera to study the bees. Okay, so it turns out the larger hornet is more dangerous to bees. This is simply the number of forager visits to a food source over which we have placed either a large hornet, the giant hornet, Vespa mandarinia, or the smaller Vespa velutina. You can see that when there's no hornet, there are about 100 visits per 10 minutes, but these decrease. Different letters indicate significant differences, so there are more bees deterred by the large hornet. Now, in colony defense, we can actually count the number of bees heat-balling the hornet. This is actually a very clever technique. I wanted to take time to mention it because often you can do very simple things in science. You simply cover this ball with a plastic bag, you seal the bag, and you open a small end of it, and the bees will fly away. and you just count the number of bees that fly away. Of course, the bees that do not fly are dead. So you know the number of bees that died, the number of bees that were in the heat ball. So it's very simple, something that anyone can do. And of course, there are more bees that heat ball the large hornet compared to the small hornet. In fact, we did a calculation, and this number of bees is exactly basically what is needed on average to cover the surface area of the larger hornet if you model the hornet as an ellipsoid. Number of dead bees I mentioned in the technique is also significantly higher. So it's a greater threat. They avoid it. They will spend a greater proportion of defenders to attack it, and is more costly. Now, it turns out that if you look, there are two different situations where bees are attacked by this hornet. One, it can attack foragers when they are feeding on food sources. And the second one is they can attack the nest where you see the heat balling. And the strongest defense, the more costly. Obviously, when it's attacking a forager, the forager would typically try to escape. You can record these stop signals, and what we noticed is they sound different. So I'm going to play you the sound of what bees will produce when they are exposed to the large hornet. Okay, so listen to the large hornet sound. And now listen to the sound of the stop signal produced when the bees are exposed to the small hornet. Okay, so hopefully you can hear the pitch difference. Higher pitch, large hornet, lower pitch, small hornet. And so this is a spectrogram, and what we're focusing on is the fundamental frequency. The fundamental frequency for the large hornet is always greater than the small hornet. And what you see is we're looking at both contexts, regardless of whether the hornet is attacking the bee at the food source or at the nest. But there is a difference depending upon the context. All right, so those are the sounds again. And here is the pulse duration. So what you find is that when the bees are attacked at the nest, you have the same pitch, but instead of the high pitch beep, it goes beep. It's a little bit longer. You can think of it as a longer scream because the animal is likely more alarmed. And this is true of whether it's attacked by the large hornet or the small hornet. So the story here is that information is being encoded in a very interesting way. The pitch tells you the severity of the attack. We do not think that this codes Vespa mandarin versus Vespa velutina. We simply think it encodes different levels of threat, and the context of the threat is also encoded, whether it's near the nest or at a food source. And it turns out that the bees react appropriately. Don't have time to go into those experiments, but basically when they're attacked at the nest, the foragers also get the signal, and they no longer go outside. They simply huddle inside the nest, and the defenders go out and try to heat ball and deal with the hornet. But how do we know actually that what I'm saying is true, that these signals actually mean anything? And so we do a playback experiment. So these are the wands with which we apply the signal to the comb next to the bee. This is because when you actually apply the signal directly on the bee, it's a bit too disruptive. But these stop signals can be applied naturally to the comb next to the dancing bee. By the way, they are always delivered more or less to the waggle dancer, with the exception of being delivered to the comb. So what you see is the number of waggle circuits per dance after the bees get a shaking signal from the small hornet or the large hornet. And you can see that a single artificially delivered shaking signal, this is one that we recorded and now playing back, and we also calibrated to make sure it's the correct amplitude, is more inhibitory when it's the large hornet as compared to the small hornet. So notice A to C as compared to A to B. Now, another cool experiment that you can do is that notice the bees don't necessarily stop right away. When they get to the stop signal, they pause their dancing, but they can resume. So what would they do if you continuously provided these playbacks until they actually stopped dancing and did not resume again? On average, 14 stop signals from the small hornet, but only 4 stop signals from the large hornet, or reflecting the large hornet, that actually stop dancing. So again, evidence that the large hornet signal is significantly more inhibitory. Now, why is this? It turns out that it's not the frequency. We did other experiments looking at this, but it turns out that the large hornet stop signal is, perhaps not surprisingly, twice as loud in terms of vibrational amplitude. Bees are hearing via vibrations than for the small hornet. So we think that the higher frequency is likely a side effect of this greater amplitude, but that in fact it is the larger amplitude that causes bees to be more inhibited by the large hornet-shaped stop signal. So why a negative feedback signal? A bee's individual experience at food can interrupt her foraging and recruitment. Obviously, that bee that experiences danger, and we have multiple experiments on this, does not tend to waggle dance when she returns to the nest. But what about the bees that are still dancing when the hornet has attacked? How does the colony get the news about this danger? And this is exactly the function of the stop signal. It provides news. We can pick up our handies, our cell phones, and get this news right away. But in the nest, we have a system that has evolved to allow the bees to understand this food source is now either dangerous or should no longer be visited. Okay, and I'll take questions at the end about both of these things. But I wanted to talk about the defense aspect, the heat balling aspect of bees signaling predators. And obviously, this is another AI image of the colony defending itself. So why is it that Apis cerana, and we know this through multiple papers, is more effective at defending itself against hornets as compared to Apis mellifera? This is a huge problem in multiple parts of the world where Vespa velutina has actually invaded. Now, Vespa velutina is wonderful to study in China, where it is native, and it is not invasive. But the wonderful thing about it is that people like to eat it. So here are some lovely dishes. It's quite tasty. It tastes a little bit like dried shrimp, or if you get the larvae fresh shrimp. And so you can buy them at the market, and you can keep them in the lab. So it's wonderful when you can get what you're studying relatively easily. This is the reason why Apis cerana is so fascinating. So you bring the hornet, and you can see about 4 centimeters, the bees start to land and immediately try to sting the hornet. The hornet is trying to sting back, and they're producing the sting venom volatile. Now, over time, the full heat ball forms, and what you can see here is there's a pumping of the abdomen, because heat balling is not only generating heat, the bees are also trying to sting. The most vulnerable part of the hornet is its varicervex, what you might call the neck. There's a chink in the armor there. They're generating carbon dioxide. They are cutting the spiracles of the hornet so that it has difficulty breathing. And so stinging, suffocation, heat generation, and increase in carbon dioxide all help to incapacitate the hornet. This behavior has been found wherever there are strong hornet predators of bees over evolutionary time. So we see this between Apis cerana and Vespa velutina. We see it between Apis mellifera cyprea and Apis mellifera caussica, these subspecies with the sympatric Vespa orientalis. We can even see it a little bit, but not to the full effective form, between Apis mellifera ligustica, the Italian honeybee, and sympatric Vespa crebo. So this occurred in apiaries around Florence, where these predators did occur. But in general, Apis mellifera has a weak, small defense and no ICU signal to the allopatric Vespa velutina. So what is the ICU signal? It turns out, and I'll just briefly mention this, the ICU signal is a way that the bees are warning the hornet that they have detected it, and therefore the hornet should not attack. And the reason why the hornet should believe this, because after all, why should you believe a bee if it's just telling you to go away, is that there needs to be a minimum number of defenders acting synchronously in order for the signal to be effective. So it's a classic example of an honest signal. The hornet believes, because the proof is right there, there are a large number of bees signaling that they can defend themselves. But for this particular story, I wanted to focus on eavesdropping. So here's the number of heat-balling bees. You can count them, again, using that very simple technique that I mentioned. All you need is a plastic bag. And what you can see is that when we provide a blank control, basically no hornet filter paper, or hornet alarm pheromone, which we previously identified and extracted from either the sting gland of the hornet or that we've chemically synthesized, because it has been identified, bee sting pheromone and live hornet. So Gray's Apis mellifera. So the only case in which the number of heat-balling bees is significantly different to the control for Apis mellifera is with the live hornet. And we think the live hornet, its appearance, its odor, and the fact that it's struggling incite a larger heat-balling response. But notice that for Apis cerana, they respond to the hornet alarm pheromone as well, of course, to their own bee sting pheromone. And this we find quite interesting because it's evidence of eavesdropping. The chemical components of the Vespa velutina alarm pheromone are not found in Apis cerana. So somehow we believe that this bee has evolved the ability to detect hornet alarm pheromone. And what would be the way to test this? So we can take the antenna of both Apis cerana, Apis mellifera. This is a GC-EAD device. You can see the antenna right here. It is electrically connected to an amplifier that records the responses of the summed neurons in the antennae. And we can pump odors past the antennae using this setup. And using GC-EAD, what we're doing is we're separating out the odors in GC. We are directing half of the flow to the antenna, half of the flow to the detector, so we know exactly which components are moving by. Now, what you can see here are the 3 alarm pheromone components that we identified, the major ones in Vespa velutina. And what you can see here is that Apis cerana is showing by these depolarizations here that it's responding to at least known nonantuone and undecantrinone. But, and this is very important, Apis mellifera essentially has no response at all. Now, it's not that Apis mellifera cannot smell this. This is at 1 hornet equivalent, which would what you would find in nature of sting. But if you drastically increase the concentration to hundreds of hornets equivalents, which would not be field realistic, these Apis mellifera bees can start to respond. But in terms of the natural level at which they might encounter it, it's only Apis cerana. And also quite interesting is if you look at this very closely, you would say, aha, there are chemical components here that you have not identified and that Apis cerana is actually sensitive to. So this is for future work. It can be difficult to synthesize for the final verification of the actual chemical. But this is very interesting. Oh, and this is the ICU signal. I'm glad I was able to get this and show you what's going on. So this is the signal that bees are producing. This is in slow motion. This is Apis cerana. And we're bringing the hornet through. And you can see they're swaying their abdomens and also moving their wings. But what's quite interesting is that this, when you speed it up and show it in a moment in real time, so we will just wait a moment here, it will appear to you to be much more synchronous as it does with the hornet. Okay, so you can see this. And it's likely that you probably have, you actually do have a very similar behavior in Apis cerana indica as well. So they're synchronized. So notice here as we sweep it by, with each sweep of the hornet, the bees are responding with a synchronous motion. And in fact, this is the start time of an ICU signal and the start time of a randomly chosen bee inside the sting cluster, and the stop time of the shaking and the stop time of another randomly chosen bee. And they are very well correlated. So again, simple experiments. What we can do, you can actually use a large cell phone for this or an iPad or similar device and do a video playback. So we present the video screen to the bees and know what happens here. We do the playback of the bee. It appears larger here, but in the actual video experiment, we had it move further away, so it's the same size as a bee. And notice, there is no threat here. There is actually no pheromone production, and there's not even an electromagnetic signal that would resemble what a bee does. It's simply only the visual signal that causes the other guard bees to produce this ICU signal. Guards are actually physically attracted and will move towards another guard bee that's producing the ICU signal, which you can see here. Here's the bee doing the ICU signal. But you might say, aha, but how do you know that the bee is not producing an alarm pheromone? We won't show you that slide, but we tested to see for alarm pheromone production. And of course, this bee does not produce an alarm pheromone. But you might say, well, but how do you know that you measured it correctly? It might produce a small amount of alarm pheromone. But in fact, it doesn't matter. The video playback does not produce any alarm pheromone at all. And they are attracted to it, and they will synchronously produce the ICU signal in response only to the video playback, and even to a video playback of a hornet, which brings us to another interesting experiment. We have the sympatric harmless butterflies that do not attack and kill bees. We have the bees themselves, and we have the hornet. And all we're doing is providing a video playback. The control could be that we have a still image, for example, of the hornet, Or we can manipulate the playback. It's very easy with any video program to change the speed. We have half speed, 1 times speed, or 2 times speed. So here what we show is that at all playback speeds, bees do not care about the butterfly. This is something they commonly encounter, and they're not producing the ICU signal. When they see the hornet, when the hornet is only doing it or moving at normal half speed for a hornet, they also tend to ignore it. But when the hornet is played back at normal speed, or even at double speed, this would be a very fast hornet, the bees are responding with increasing the number of ICU signals. Now, what about the bee here? At half speed, they don't care. At 1 times speed, they do care, and they respond with ICU signals. But at 2 times speed, they discriminate. They think, this is an anthropomorph saying it, this is not quite right. This is not an ICU signal. So they're very selective, and we know that they are simply visually going off of how the potential threat is moving. So relevant video playbacks trigger the ICU signal. And this is very important. You're probably familiar with the story of the boy who cried wolf. How do you, especially in a social situation, discriminate between honest signals and signals that are either dishonest or incorrect? And this is very important because, after all, a bee seeing another bee producing the signal triggers a cascade of bees clustering and trying to defend the colony. They do not actually have to see a predator. They're simply trusting each other that this guard bee has actually seen a threat. And the reason we think that it has evolved this way is that in order for the signal to honestly communicate to the hornet that the colony can heat ball, weak colonies cannot effectively heat ball or deter hornets, it has to be rapid. So it should be contagious. It rapidly spreads throughout the population. It rapidly recruits other guards, and they move in synchrony, possibly because this creates a larger optical illusion, this hasn't been tested yet, that there are more bees there than there actually are, but also that it shows the colony is capable of coordinated attack action, a large number of bees to heat ball. So first step is hornet detection. The bee sees the hornet. The bee starts to immediately produce the ICU signal. This visually attracts other guard bees. The final component is that all the guard bees immediately start producing the ICU signal for it to be contagious and synchronized. And therefore, that is why in that initial image, the hornet is departing, because it honestly detects there's no point in attacking this colony. It's too strong. Time to move on to a weaker colony. So I just wanted to thank my multiple collaborators, Dong Shihao, Tan Lin, and Tan Wenping, my lab, the bees, and the different funding agencies. And I'll just end by concluding that just in these two little stories, I hope you can see two things. One, that you can do very simple experiments to study these bees. You don't necessarily need high technology. In one case, all you need is a plastic bag. And that you can learn a great detail about the predator and prey interactions when we discuss the fitness landscape. This is a concept that equally applies to animal behavior. So thank you very much, and I am happy to take questions.
Thank you so much, Professor Nieh. That was very, very interesting. So I was just wondering, so you did say it's a honest signal, and I get it, but is there any chance for cheating to happen? For example, in a weak colony, if they show enough strength, but technically they are not really strong enough to handle the hornet, but do they sometimes take the risk and clump up in large numbers? Because if they scare away the hornet enough, then they don't really have to end up taking the risk. So are there instances where they actually do that, even if the colony is not strong?
So in our observation colonies, there are typically only 2 combs. They're much weaker than a regular colony, which would have 10 combs.
I see.
And what we find is they continue to do the ICU signal, and they muster as many guard bees as they can, because these hornets are a very severe threat, especially when they attack the colony. Only a few hornets can actually kill all of the adult workers within a day, and then they go in and they get the larvae, which is what they actually want to bring back to their colony, because of course they're also social insects. So they're both operating at the limit of what they can actually do. And so although a weaker colony would send out a large number of bees, a stronger colony can do even more so. And therefore, I think that's where the honest interplay occurs.
Right. And just what's cool with that? Do the hornets ever evolve in the sense whether they start recognizing if they wait enough, they'll get to know the real strength of a colony? And also, if it's just one, I know hornets do not have much of a communication, or rather, they do not have the kind of recruitment process that bees do. But if, let's say, a couple of more number of hornets come, it's not really a good news for the bees. So do they ever sort of learn from experience and sort of, start communicating to each other? And have you ever seen instances like that?
So I think the way in which hornets, which are also highly social, are recruiting other hornets to attack a food source, the honeybee colony, is very interesting. What we know is that they have marking pheromones that are produced abdominally, and they can mark the entrance of the colony. You may have seen a paper by the group that is working with the Vietnamese Apis, and also their house hornets, where they use animal feces to kind of overmark or cover over the odors of the hornet pheromone. We do know that the hornets fly around in our apiary every day and are checking out the Apis mellifera and the Apis cerella. So they're flying around, and we do see they tend to attack the Apis mellifera more. In a separate experiment, we found that most of the Apis mellifera colonies actually died at the end of this experiment because they were not able to withstand these constant inspections and attacks.
Right. Actually, we have seen that here in our colonies as well, where actually a lot of hornets start coming in groups and start attacking mellifera, and mellifera colonies cannot withstand it ever. So I remember once when Guard Otis had come here in Bangalore, and he did show the videos of these Vietnamese colonies where they had smeared the entrance of their colony with animal feces and waste and anything that they could find. And that seemed to sort of deter the hornets a lot. So yeah,
it's quite interesting. There's a paper about the filth gathering of bees, which I encourage you to look at. Sorry, go ahead. There was a question.
No, actually, we'll take audience questions in a bit, and you can just type in your questions, and I'll read it out for you. So hopefully that will help. Sorry, continue, Doctor.
Okay, yeah. Wonderful. So I can read it. Sorry, thank you. And I'll just say on this that there's fecal gathering in stingless bees as well, which you, I believe, also have in India. And so I think it's a fascinating subject. If anybody wants to study why they do it, it could be interesting.
Yeah. No, totally. I do agree. We do really need to look more into it. Actually, Professor Belavadi, I was just wondering, so in these sort of different behaviors that you see in terms of pollination for cardamom versus muskmelon, so how do these, sort of studies and behaviors sort of help form, the policies that we have for agricultural practices? And
with reference to cardamom, in fact, we have actually worked out the number of forages required per hectare like that by a number of,
okay.
Yeah, because in 1 hectare there will be something like 3,000 plants, okay, and if each plant produces 20 flowers every day there will be about 60,000 flowers. So we have calculated how many bees are required to sting and all that. So we ended up with 2 colonies of Sarana per hectare like that, and in fact this has gone as a recommendation also. Yeah.
Wow.
In fact, that definitely helps this kind of work.
Yeah, totally. Yeah, because with this way, you can actually advise the number of colonies that are required. And yeah. And, so I was also wondering because, so in muskmelon, it was Floria and Sarana, whereas in cardamom, it was Dorsetta and Sarana. So I was just wondering, Sarana is like a common concept here. Do you think they evolved because we have domesticated them and we keep introducing them?
We also have a lot of feral colonies also. Sarana feral colonies also are there. Plenty of colonies will be there in the field, especially in the area where we worked some of this work. Dorsetta colonies, in fact, I should say, are dwindling in some places because we have seen that in many places they have cut down the huge trees and all that where the Dorsetta would nest. And I think there's a lot of work that has been done recently in the urban areas also about Dorsetta colonies.
Yeah. So is there any concrete studies to sort of look at how much is the decline in Dorsetta? Because that will help sort of info,
I think
because
there's some study done by A-Tree.
right, yeah.
Yeah, they have done some work on the number of colonies over years, how it is coming down. For example, in our own campus in GKVK, we had huge uh, trees which used to harbor nearly 200 or 300 colonies at a time.
Oh, yeah.
Now, because of that disturbance, human disturbance, now we find hardly 10 or even less number of colonies there. So this is the thing. It is human disturbance is too much affecting the colonies also.
Yeah. I totally understand. Speaking of Dorsetta and Floria, Professor Nieh, I was just wondering, so we don't really hear a lot about hornet attacks on Floria and Dorsetta. Although I'm sure that is also happening quite a bit. It could be that, we are less interested because they are not domesticated as such. But I was just wondering if, do you know of any studies, or if you, in your experience, have seen any sort of maybe different strategies that they use? Although with Dorsetta, I can sort of imagine the threat is maybe not as huge, but still, I'm wondering.
So we've done some work on Dorsata and Floria with predators, hornets and ants, and also looked at the different kinds of vibrational signals. In both cases, they produce something which, in response to attack, is similar to the stop signal that we heard by Apis cerana. With Floria, what happens is that they seem to vibrate the entire comb. As you know, it forms a small comb. And if this continues, if the attack continues, they abscond.
Yeah.
So typically the defense of the Floria colony, we believe, is that it forms within very thick vegetation. And if you worked with it, you know that they don't like it if you remove the thick vegetation because it is fun. But yeah, their ultimate response is to abscond. We did these experiments inside a tent, so they would abscond, but they had no place to go, so they had to eventually come back.
Oh, no.
And Vespa tropica, it attacks Rhopalidia colonies, Rhopalidia wasps.
Oh, yeah.
Yeah.
Yes. I know, because I used to...
Rhopalidia had no defense. Rhopalidia, the workers of Rhopalidia, they just go and then hide behind the nest, and then this Rhopalidia lands on the nest and then takes out all the pupae, larvae,
all the pupae.
falls off.
Yeah. And in Rhopalidia, they don't really attack the individuals per se. They only eat the larvae.
Yes.
Maybe because they are fellow wasps. It's I'm guessing there must be certain things that is not edible. But yeah, I have seen, because I used to, for my PhD, work on Rhopalidia, and I've lost numerous colonies for to Vespa attack. Yeah, they have no defense at all. They would just sit in a corner and just watch a hornet actually. Yeah, which is really fascinating. Yeah. I'm surprised they've not developed by efforts till now. Yeah.
I was going to just end by saying that for Dorsata, we also see something similar to a shaking signal. Sorry, a stop signal. I think the main limitation is for us, we have limited access to Dorsata. It is a bit inaccessible. It doesn't, in Kunming, it doesn't nest.
Right. And also, I'm guessing the sheer size of the colony, like an individual Dorsetta is also big in size, as well as an entire colony is huge. So I'm guessing they form a much bigger threat as well to the hornets. So
they do, and they have what's called this Mexican wave, where you can see the ripples spreading throughout the colony.
Insect types.
Yes, and then that we think is also an example of a contagious and honest signal to the predators of the size of the colony, and of course that it's been detected.
Right. Yeah. Thank you. I think since we are running short of time, we'll take a few questions from the audience. So I'll just check for the questions that's been put up. Actually, Zachary Huang has commented that he tried moving his hands and the bees, basically Serana, will respond that after a wasp, they will respond in a manner as if a wasp was already there, it seems, when he tried moving his hand. So it's a comment.
Mm-hmm. And I also commented back that they can do this for predators such as bears or for humans. What appears to be important, we're still working on this, is that the wasp is moving in a way to check out the entrance of the colony. So horizontal motion. We are expanding our video playback experiments to figure out what is it that allows a bee to distinguish between a wasp or a butterfly.
Right. Yeah. Visual cues, I think, are pretty strong here.
Yeah. Mm-hmm. It's quite interesting.
Yeah. We have another question. So someone asks to Professor Belavadi, how do they know about the nectar availability without landing? So I'm guessing they mean the cardboard ones. Is it with vibration or smell?
Yeah. In fact, when a bee lands on the flower, it actually leaves some marks there. That is, it's called as the footprint pheromone. No footprint pheromones are there, and that is highly volatile, and that will be gone in a matter of about 10 to 15 minutes. So during that time, there will be secretion of nectar. Nectar accumulation will be there. And then after 10 minutes, if another bee comes, it will not be able to find out that a bee has already landed there, and then it lands there and then gets a little bit of nectar. So that's how it is. Yeah.
Wow. Very fascinating. Time enough to regenerate the nectar.
Exactly. It is very interesting. Yes.
We have a question to Professor Nieh from Hiren Gogoi. He says, can we have some alarming pheromones to deter hornets?
So, that's interesting. The hornet alarm pheromone, if you were able to synthesize it, might deter other hornets from coming to visit that particular food source. But I suspect that it's something that they would probably become accustomed to if a beekeeper were to keep them in large quantities. I know that what tends to work for yellow jackets and other hornets is that beekeepers can restrict the entrance size of the colony. In the extreme case, the hornet is much larger than the bee. So just like a queen excluder, the hornet could not enter the colony, but the bees could come out, although this would have a cost in limiting foraging.
Yeah, that's true. We have a question to Professor Belavadi. So Austin asks, why would a plant, say like cardamom, produce more nectar than is what is used? Isn't that a waste of resources?
Exactly.
And is there any reason they have tubes longer than the bees' proboscis?
The thing is, the honeybees are not the original pollinators of cardamom. The original pollinators of cardamom are Amegilla species, which have very long, proboscis, and they can exhaust all the nectar. But the honeybees in the cultivated systems, honeybees actually replace these. They do not allow, they get disturbed. The Amegilla get disturbed, and so they stop visiting the cardamom flowers. And the honeybees, they take over the whole thing. And interestingly, our cultivation of cardamom, in fact, we have given an input to the breeders to look at short corolla tube varieties. And in fact, all the high-yielding varieties have shorter corolla now. So that is the thing. So shorter corolla means so much of nectar so much of energy is saved. So that is the...
Right.
That's all. Yeah, that's a good question.
But wouldn't that sort of end up, deterring the original pollinator? Wouldn't it, eventually?
No, because in the cultivated system, we are talking about the cultivation of cardamom. In the cultivated system, when bees are the major pollinators, where we can actually manipulate the bee colonies and all that, and depending on bee colonies, we do not need such long corolla tubes. In fact, we looked at a germplasm collection of cardamom varieties in different institutes, in cardamom research stations, and we have measured the corolla tube length of about 145 entries, and we have found that it is in fact there are some varieties which have some entries which have something like 35 millimeters long corolla. We just wonder what might be pollinating that in the wild.
Yeah.
Wow. Don't know what.
there are so many species that we have don't even have any idea about, the amount of pollinators, the wild bees that's out there. Wow. So, Professor Nieh, it's a very bit of a general question. So till now, what are your limitations? Is the question.
I think that one limitation, we would like to study this in many different other kinds of bees, Kashevna colby, nuluensis, is actually getting those species in abundance so that we can study them. For dorsata, I think it's quite fascinating. And there are certain places where they are nesting more commonly. We actually went to Thailand to temples because near temples, they still have the large trees and they are allowed to nest. So getting access to the species, I think, has always been a challenge.
Yeah, I can imagine. So there's a question which is a more general question, which I will leave to you guys to answer. It's a it should be interesting. Do bees forage during the night? And if yes, how?
Yes, they do.
Which is, I know, a huge topic.
Especially Dorsata. I think there is work done by Axel's students and Axel's group. They do forage in the night. And there are some species of xylocopids also, which actually forage in the night. Yeah.
Mhm. And they also ask if yes, how? I know that's a long answer, but
I'm not very sure. They say that they actually do not use the position of the moon or anything for recruitment and all that. They actually still will be using the position of the sun. I'm not very clear about it. I think there is information available on that. You can maybe he may add to this.
yeah, actually recent work in our lab itself. Yeah. Sorry, Professor Nieh, go ahead.
Oh, no, I was going to say that actually you would likely be best to talk about what they're orienting to, whether it's the last remembered position of the sun or the moon. But what's quite interesting is that many bees, including the Megalopta, have the ability to see under very low light. For the Dorsata, it's not as extreme as the Megalopta, but apparently they will fly out and forage on moonlit nights when there's relatively bright light. And so I think the question of how is related to their visual sense.
Yeah, which is true. And it's also seen that Dorsata seem to have a larger ocelli compared to its counterparts, which might be also helping it with its visual acuity in the night. And yes, you're right. With the recent work in our lab as a student, we've seen that they seem to be orienting to the last position of the sun and not really the moon. So, yeah. Hopefully you'll read more when the paper gets published. So Axel has a comment. So, yeah, definitely, yeah. So Axel has a comment to you, Professor Nieh. He says that I agree with him that the EAG recordings for the hornet pheromones do not mean that Serrana has an olfactory receptor gene that Apis mellifera does not have, but that it likely indicates a difference in the number of sensory neurons expressing that receptor. So transcription studies would be sensitive enough to show that difference. And in addition, it could help to identify the receptor gene. So it's a comment to you.
Thank you. No, that's a great point, and it's something that we would like to do to explore this question in great detail.
So he also has a comment to Professor Belavadi. So he says, I was very impressed by the complex provision of nectar and pollen by the plants. Such studies are important to provide a picture of what complicated cognitive decision bees have to do in nature. So yeah, that was a comment. I think we'll end with just one or two questions. So we have a question from Bharat. He says, can guard bees signal the returning foragers about the hornet? This is to you, Professor Nieh. Yeah.
So the guard bees would only produce the ICU signal when they've actually seen the hornet threat, or possibly when presented by the hornet alarm pheromone. But they have no way, we think, of predicting when the hornet would come back. But we do know that this ICU signal only actually works at times of the year when hornets are present. So in the winter, when the hornets are not there, you can do the same visual playback, but we find very little response in the bees. So they must have a way of knowing when the colony is likely in the season to be attacked by the hornet, and therefore they produce the signal appropriately.
Right. I'll end with one last question to you, Professor Belavadi. There's a question by Sangeeta. She asks, is there any nectar robbing phenomenon happening in cardamom flowers?
I have not actually seen, but there are some reports of nectar robbing, but not by the honeybees. I think some solitary bees do that. Yeah. And I have not seen that. Yeah.
Okay. So I think if there is no further comments from the both of you, because we've overshot a little, I think we'll end this webinar. Thank you so much for being here. Both of you have really interesting talks, and the discussion was also very interesting. Thank you so much for accepting the invitation.
I went fast. I think I...
Oh, no worries, sir. Yeah. But it was quite clear. So, yeah. Yeah, thank you both so much. And before I end, just in case any of you would like to support CWS initiatives and other similar outreach work that we do, you can visit our website at cwsindia.org or contact us at outreach@cwsindia.org. So with that, thank you everyone for joining in and making this wonderful discussion. Yeah, thank you both.
Thank you, everyone.
Yes.
Thank you.
A little bit about what CWS is. Center for Wildlife Studies is a 37-year-old non-profit organization specializing in wildlife research, conservation, policy, and education. Now, until 2020, we used to host public talks on the latest topics in wildlife science and conservation in our Bangalore office. Now, due to the pandemic, we have instead been conducting a series of webinars. So far, our webinars have covered a wide range of interesting topics, such as public health and zoonotic diseases, tigers and elephants, snake bites and bioacoustics, and today's social insects. So you can visit our YouTube channel if you want to view our previous webinars. Today's webinar will also be recorded and available on YouTube.